Okay, so this is going to be chapter 12, nervous tissue. And this is really the first time I'm going through this new strategy of scanning over the material once, just like reading the chapter section titles, looking at facts and figures and stuff, which takes like 2 minutes. And then reading through the material, making highlights, jotting down some notes, in the book, in the iPad, trying to do some active recall here and there. Then I go over the material a second time and I write out what I call story notes, which is this document that I'm about to read. And that's really revisiting that material for a second time, really fleshing out some ideas. Okay. And then after that is actually going to be testing myself in some way. And whether that's finding a test bank for this chapter, whether that's finding a Quizlet for this, taking an exam, and rather than going through a selection of 50 questions, doing the entire bank of 150 and seeing what I remember and learning from that. And that's really based on this new thing that I read, that you learn more, like exponentially more, from taking an exam than studying, than any other form of studying. So I'm trying to really commit the fundamentals of A and P to knowledge. And then the very last step of this process is a video, is making this video and reading through, and then, after testing myself on it, being able to teach it. And hopefully that's a really good capstone of an entire chapter and committing this to memory. So, all right, without further ado, we're going to dive right into it. This is the first of, I think, 5 or 6 chapters on the nervous system. And this is chapter 12. This is nervous tissue. Okay. So the nervous system has anatomical and functional divisions. All right. So I'm just going to go right through to what you want to kind of remember here is you have the central nervous system, which is just the brain and spinal cord. Then you have the peripheral nervous system, which has a lot of subsets to it. So the two big ones that you need to remember are the afferent and the efferent division. Now, the afferent one, that's the simpler one. That's just sensors and receptors that are going to be sending information. Okay. The efferent division is where everything else is located. So this not only has a somatic nervous system, and this is like moving your arm, so things under your conscious control, but also subconscious control. So I like to think of this as if you reach and grab a hot frying pan, that's part of your somatic nervous system. And then the reaction of you dropping that frying pan, which I believe actually goes to your spinal cord, it doesn't even get to your brain before your hand is already removed. That's all the somatic nervous system under the efferent division of the peripheral nervous system. Okay. Now, the autonomic nervous system, this is all of the subconscious stuff that you're not even aware of. Like, you're aware of grabbing a frying pan and dropping it, but this is like your breathing, which you're now aware that you're breathing. This is like blinking, your heart rate, blood flow, all the internal stuff that's going on. That's automatically happening. Okay. Now, it itself has the sympathetic nervous system and the parasympathetic nervous system. This is kind of like that fight or flight complex. Sympathetic is, it's going to like dilate your eyes. It's going to speed up your heart rate. It's going to speed up your breathing. It's going to get you ready to fight. Parasympathetic is going to slow things down. It's going to slow down your digestive tract. It's going to like constrict your pupils, lower your heart rate, things like that. Okay. And then there's the enteric nervous system, which they really don't talk about, which technically is part of the larger umbrella of central and peripheral. So just kind of know that that's there. Kind of a mnemonic for the peripheral nervous system is just, it's F and F. The F is just like the sensors. And then the F is, I have sum and auto. So sum is like somatic, and that's the smack and pull a stove. And then auto is that go and slow. All right. So this is this in kind of picture form. So yeah, you have to drill down pretty deep to get to the parasympathetic and sympathetic division, which I feel like you hear a lot about. All right, so now we're going to go right into neurons, which is the basic functional unit of the nervous system. And here's a picture. right here. So this whole thing is the neuron. You can see the image here. We have the axon, which is this long arm here. We have the cell body with the nucleus. The nucleus is surrounded by something called a perikaryon, and that's a special name for the cytoplasm surrounding the nucleus. All right. The axolemma is the name of like the plasma membrane itself. So the neuron has a special name, and it's specifically of, I believe, the axon itself. If you have something that branches off kind of in the middle of the axon here, it's called a collateral. This kind of converging point where the cell body narrows into the axon is called the axon hillock. And at the very end, you're going to have the postsynaptic cell. So this is the cell body. The interesting thing is the dendritic branches, it doesn't show those on this other cell. I think those connect to like other neurons that aren't necessarily involved in the action potential that goes through like this main axon here, but they're not a receiving center because the receiving center is right on the cell body itself, as you can see here when you have these axon terminals, which we'll get to in a second. But it's the telodendria. That's this end point here, not the dendrites, not the dendritic branches. That's what you want to remember, okay? Speaking of the dendrites, they actually do make up 80 to 90% of a neuron's surface area because they're so branched. So even though we don't really talk about the function that much, and we do talk about the telodendria and the axon terminals and the synapses, just keep that in mind, all right? We also have, so you have to know that the perikaryon, this area around the nucleus, has RER, which I had forgotten was rough endoplasmic reticulum, and it has these free ribosomes, which of course are going to make proteins. These regions that have these, the rough ER and the ribosomes, are called Nissl bodies. So just something you have to remember. All right. So we can break the classification of neurons down into based on their function. So either sensory, motor, or interneurons. So the sensory ones, also known as afferent neurons. And again, if we go back to the afferent division, these are sensors and receptors that are kind of collecting and sending information. They're found in ganglia, which are like these clumps of neurons. We have about 10 million of these, and they collect information from either the internal or the external environment. They themselves are either somatic or visceral. So visceral are the internal, and somatic are external. We can also kind of alternatively look at sensory neurons by these three classifications: the interoreceptors, the exteroreceptors, and the proprioceptors. I did see some test questions on this. So the interoreceptors, it's your inside stuff, your digestive, respiratory, cardiovascular. The exteroreceptors, this is a good one to remember. It's like, it's not your 5 senses, it's just 4. I believe it does not do sight. Oh, it does do sight. Sight, smell, touch, and taste. So that's what exteroreceptors are doing. It doesn't do pain. Interoreceptors, they're the ones that do pain. So that's something you want to remember. And then proprioceptors are like the position and movements of your muscles and joints. So like my arm is above my head. That would be proprioceptors in my arm and my hand that are keeping that in mind. All right. So that's sensory. Okay, now we're going to get into the motor neurons, which is, these are going to be in the efferent division. So again, the efferent division is the smack a stove and pull your hand away. All right, so more of like the effects, more of the actions here. So we have a 500,000 of these, so not nearly as much as the 10 million sensory neurons we have. So just remember, we feel a lot more than we actually affect and move and act on. All right. The sensory neurons are collecting information by being affectionate. This is kind of how I remember that. And then they send this information to the CNS to process, and then they make an effect through the efferent division. All right. autonomic nervous system. We have these visceral motor neurons, you know, et cetera. We have these interneurons. So we said there are 10 million sensory, 500,000 motor, but there are 20 billion interneurons. These are located between the sensors and the effectors, the motors. And these mostly are in the brain and spinal cortex. That makes sense. And these are also involved in higher end functions such as memory, planning, and learning. So they're just involved in everything. They're just kind of everywhere, almost kind of like the glue that holds everything together, I guess you can say. All right, so that's the neuron and just some of the classifications there. But we also have these support cells. So we have, let's see if I can remember some from memory. The CNS and PNS, central and peripheral nervous system. The support cells are called neuroglia. Hope I'm pronouncing that right, neuroglia. The central nervous system, we have these astrocytes, which make up like the extracellular matrix. We have oligodendrites, which I believe are involved with myelin, which we'll see is this this wrapping, this coating, this insulation that go over the neurons. We have the, I think, ependymal, which have to do with cerebrospinal fluid. And then we have the, I think it starts with an M. They're like the janitors. I think they're just called microglia. And those are like the cleanup crew. So let's see if I got all that correctly. So astrocytes, ependymal, oligodendrocytes, and microglia. Yeah, so the astrocytes are the largest and most numerous. They maintain the blood-brain barrier, and they repair damaged nervous tissue. Okay. They provide structural support as well. The ependymal cells, they produce and monitor the cerebrospinal fluid. The oligodendrocytes, yes, are these guys here, and they kind of have these legs that turn into these myelin sheaths, which is this insulation of the neurons. All right. They insulate and speed up action potentials, which we'll get to shortly. The area where there's no myelin are called nodes. You can see that in the blue here. And the myelinated areas are known as the white matter of the nervous system. All right, we'll look at the bigger picture in a second. Like I said before, the microglia are the janitorial service and police force of the nervous system. Let's just see here. Could be microglia. Microglia. Okay, whatever. All right, so now for the bigger picture. See what I did there? So here are the ependymal cells, kind of these, what are these? Cuboidal epithelial cells. Simple, definitely, because they're in one row. And so, yeah, there we go. Simple cuboidal. I swear I did not look at that before just guessing. They line the fluid-filled passageways to the central canals where you would have cerebrospinal fluid. The microglial cells, so these are the janitors, police force. They remove unwanted substances. The astrocytes are kind of the main guys, and they are kind of creating this blood-brain barrier. See, this is a capillary here, and this is kind of like the intermediate that's taking, I guess, some of the oxygen and nutrients from the blood and feeding it into the nervous system. But it's going to be selectively permeable, which is why the blood-brain barrier is so important in pharmacology. All right, then we have the oligodendrocytes, which are wrapping these neurons in myelin. So this is all pretty cool. And remember, this is the this is on the central nervous system. So this is all brain and spinal cord that are involved with all of these. So just know these four neuroglia and their, you know, basic functions, how they differ. Okay. There are two neuroglia of the peripheral nervous system. These are the satellite cells and the Schwann cells. And I believe the Schwann are the ones that wrap around and create myelin in the peripheral nervous system. The satellite cells, they also surround neuron cell bodies. They're not talked about much, but just know that that's what they do. The Schwann cells are the ones that have to do with myelin, and they form what's called a neurolemma. So not the axolemma, but a neurolemma. So another type of cell membrane, specifically of the peripheral nervous system axons, all right? Neurons can repair themselves more so in the peripheral nervous system than in the central nervous system. So think about damage to your brain or spinal cord. That's going to be a lot more significant than, nerve damage anywhere else in the body, your arms, legs, things like that. Okay. All right. Now we're getting more into the meat and the potatoes of this chapter. We have the membrane potential of a neuron is determined by differences in ion concentrations and membrane permeability. So I've been working on this section for like over a year off and on. Okay. I'm going to skip a lot of what I wrote earlier, and let's talk about a few of these central tenets or commandments of the normal resting membrane of a cell, which ultimately is going to look something like this. Okay. Here's the deal. This is what you need to remember. We have more potassium ions inside of our cells compared to sodium ions that are on the outside of the cells. I like to think of this, that we are, we keep ourselves very well fed, full of nutrition, and our cells are full of bananas. They're full of potassium, okay? And we keep the salt on the outside of the cells. We don't really want the salt from a nutrition standpoint coming into the cells. So that's how you'll remember there's more potassium on the inside. But you also want to remember that we are very sad and we are very depressed on the inside, because the inside of the cell is more negatively charged than the outside of the cell. And I kind of drew that here a little bit. So we have mostly negative because of, well, we'll get to that in a second. These are kind of slightly unrelated concepts. And on the outside, we have more sodium. The reason that the inside of the cell is more negative is for two reasons. One, I didn't draw this here, but there are, here we go, big negatively charged proteins trapped inside of the cell. So big negatively charged proteins trapped inside of the cell. And potassium moves out of the cell faster than does sodium coming into the cell. And these are both positively charged cations, right? Positive cations. Took me a second. Anion is negative. So these are positively charged cations. They're both in the same column of the periodic table. So they're just a +1 charge here. But more potassium is going out. So there's more positive on the outside. And you could see that they have these little, they have them right here. And then there's more negativity on the inside. So just to refresh what we just learned here, we have more potassium ions on the inside of the cell, period. We have more sodium ions on the outside of the cell, period. Okay. They're both positively charged ions. Don't get that confused, but there's more potassium on the inside. In other words, there is potassium is moving out faster because of the concentration gradient. Okay. Yeah, so potassium inside, sodium outside. Potassium is moving out faster. Sodium is moving in as well, but not as fast. And then those negatively charged proteins on the inside, in addition to the fact that the potassium ions are moving outside faster, that's why we have negative on the inside, positive on the outside. Okay. So it's kind of complex. You kind of just have to like work through it, stare at this picture for an hour and just figure out what's happening. Now, here's the deal. And I'm just going to read my description here. One of the basic rules of fluid mechanics is that ion concentration through a membrane will even out. Ions move from areas of higher concentration to areas of lower concentration. The cell membrane has two leak channels, like a shallow slide or a waterfall leading from one pool into another pool. And in this case, that's the cytocell to the extracellular fluid in potassium's case, and from the extracellular fluid back into the cytocell in the sodium's case. This process through a leak channel of ions trying to reach equilibrium is called a chemical gradient. So it's the most basic garden variety gradient. It's simple chemistry of ions going from an area of high concentration to low concentration in order to even themselves out, okay? So yeah, potassium moves outside more quickly than sodium. Okay, but here's the deal, right? If all of the potassium on the inside are taking the water slide out of the cell, where there's less of them, and they're going along that chemical gradient, and sodium is doing the same thing. They're from the outside of the cell coming in where there's less. If that just continued to happen, you would eventually reach equilibrium and nobody would be moving anywhere, and the chemical gradient would stop. So we have the sodium potassium pump, and enter one of the most infamous mnemonics in all of pre-med, which is 321 Nokia. What this stands for is 3 NO is sodium out, Na out, 2 Ki, which is potassium in, for 1 molecule of ATP, which we're not really getting involved in with ATP here. What this does, and don't get too in the weeds, I tried to understand why are we sending more sodium out and only bringing 2 potassium in when it sounds like we would want to be bringing more potassium in. I tried going down that rabbit hole. I could not reconcile it. So don't really pay attention to why it's 3 and 2. Just know that it's 3 and 2. Okay. And what happens is the sodium potassium pump allows this phenomenon to continue. It allows all the potassium on the inside to keep moving to the outside because it's bringing everybody back. It's kind of like either an escalator or an elevator or a ladder that allows these ions to get back to their side of the pool so that they can take that slide down again. Okay. So the sodium potassium exchange pump is exactly that. It's like a little elevator that requires energy to run, or it requires work to climb up a ladder. So that's how you'll kind of remember that ATP, but it prevents everybody just forming equilibrium and everything stopping. Okay. So keep that in mind. All right. The other thing too here you want to remember is that potassium, I may have misspoke earlier, potassium moves out quicker because the membrane is more permeable to potassium. All right, membrane permeability for potassium is greater than sodium, hence the bigger arrow here. So there's a kind of a thicker arrow here, and there's a thinner one with sodium coming down, okay? 2 numbers you do want to memorize is that the equilibrium potential of potassium is -90 millivolts, and the equilibrium potential of sodium is +66. Okay, so that's the chemical gradient. The electrical gradient, because more potassium is moving out, we have more of a positive charge outside of the cell. Remember, we're sad on the inside. Also, because we have these big negatively charged proteins. This creates an electrical gradient. So this is kind of an aside. The chemical gradient is just these leak channels, these water slides. The electrical gradient is kind of like the bigger picture of we have all this negativity on one side, we have all this positivity on the other side, okay? And it turns out that's about -70. That's our threshold of our resting membrane potential. Now, if you are comparing the electrical gradients, the electrochemical gradient is the combination between the chemical gradient and the electrical gradient. The electrical gradient itself looks, the arrows look about the same, but the net electrochemical gradient is much different. The net electrochemical gradient for sodium is much larger. I think because you have all of that positivity on the outside being attracted to all of that negativity on the inside. And positive is going to go towards negative. So when we are ready to let more sodium in through what we'll learn about are these other channels, it's going to be a much bigger impact. Sodium rushing to a cell causes what we'll learn in a second, depolarization, action potentials, and all of everything, basically. All right. That kind of just clicked for me. But you can see the potassium here, positive, we're just moving to an area of positive. So the net electrochemical gradient there isn't as nearly as significant. Okay. There's also like this fish metaphor. Let's read this in a second. Let's read this. So they hit it out of the park with another great metaphor. The cell is a leaky fishing boat full of caught fish. As we go through the ocean, water floods in. As we go through the ocean in a boat that has holes in it, water floods in and we lose fish as they swim out through the leaks. This is where the sodium-potassium pump comes in. It bails the water out by ejecting 3 sodium ions and recatches fish by bringing in 2 potassium ions at the same time. Okay. At normal resting potential, the pump ejects sodium ions as quickly as they enter the cell, so it exactly balances the passive forces of diffusion, and the resting membrane potential remains stable. So it allows those 2 water slides to operate. Okay, and if there's like one thing you can remember here, it's just that like the leaky potassium channels are kind of more important because they're moving out faster, and that's pushing the positive charge outside of the cell, and it's sort of helping maintain the negative charge inside of the cell along with the negatively charged proteins. That's something I remember from earlier classes. All right, so let's go into the action potential here, which I didn't get into a lot of detail because this topic is just really done ad nauseum. I'm kind of sick of it personally, but I did talk about it a little bit here. All right. So a stimulus opens up the sodium channels, and sodium rushes into the cell. That depolarizes the membrane, which is this part between 1 and 2. So you have to hit that stimulus first for things to start happening. And remember, depolarization is, even though D is kind of a negative, is increasing our threshold. It's increasing our threshold up above 0, okay? The depolarization reaches a limit, and it triggers the sodium channels to close or inactivate. That's when we talk about this absolute refractory period where the cell, the neuron, cannot accept any more stimuli, okay? The voltage gated potassium channels open, and these voltage gated channels are exactly what the name suggests. When there's a change in the membrane potential, they are triggered to open. All right. And it's kind of a check and balance system that, oh, all of the sodium just rushed into the cell. We need to get some positive charge. There's way too much positive charge into the cell. We're going crazy. We need to get some of this positive charge out. So the potassium ion voltage gated channels are triggered, and they say, get some potassium out of here. We need to push some of this positive charge out. And that's exactly what happens. So you could see, and that follows the charges. We're going positive, positive, positive. The cell's getting all of this positive charge in. We hit about plus 30, no more. We hit this absolute refractory period. The sodium channels are all cut off, and they are triggered by this change in voltage. They're cut off, and then all of the sodium, all the potassium can now start being pushed out to then correct this. And that's what's called repolarization. And we'll see between 3 and 4 here that the millivolts are going back down into the negatives. We actually go down a little bit too much. That's called hyperpolarization. All right. And then we will even out a little bit into what's the normal threshold again. Okay. And that's what's called the relative refractory period. So the neuron isn't quite ready to accept a new stimulus. If it can and it's strong enough, it will depolarize and it will go through this whole thing again, okay? We do have continuous and saltatory propagation. Continuous is with an unmyelinated axon. It's like whispered down the lane in a queue of people where the story is retold at each person. So distance doesn't affect the message. Saltatory is similar, but the people are separated by like 10 feet. So you can get the message across a lot faster. It's kind of like a subway system of going from stop to stop. You're kind of going at one, shooting to the other, and then shooting to the next one. It's like a bullet train, all right? Axon diameter is a thing. There's something called type A fibers, which are the thickest, and they can send signals up to 268 miles per hour. That's happening in your body right now. That's crazy. All right, let's talk about, I realized I didn't even have the term like hyperpolarization. Okay. All right, we're getting there. 3 sections left. Now we're going to talk about synapses. We have electrical and chemical. Electrical are very rare. These are direct contact between the cells. That's really all you need to know there. Chemical synapses, these are the ones that we hear about all the time. These are the narrow spaces between 2 neurons, which is called a synaptic cleft, which we can see right here. So this is the presynaptic neuron that is sending the message. This is the postsynaptic neuron that's accepting the message, okay? Axon terminal, the presynaptic neuron is full of mitochondria and tons of vesicles that house the neurotransmitters. So we can see the vesicles here with acetylcholine. With electrical, the signal is always propagated. With chemical, that's not always the case. You could send a message to a synapse, but if you don't have enough to kind of like push things over a threshold, then nothing's going to happen, okay? Our first neurotransmitter that we're going to talk about, a neurotransmitter it's a signaling molecule, all right? And the first one is called acetylcholine, which is shortened to ACH. If you have a synapse that releases acetylcholine, it's called cholinergic. So not acetylcholinergic, which would be a much better name in my opinion. But I think technically because choline is separated, but whatever. I have been studying some pharmacology. So dicyclamine or bentyl is an anticholinergic used to treat diarrhea and IBS. Acetylcholine is the most widespread and the most studied neurotransmitter. It's found at all of our neuromuscular junctions involving skeletal muscles, which are being used to for me to speak and to type that sentence. There are also synapses in the CNS, our brain and spinal cord, all the neuron-to-neuron synapses in the peripheral nervous system, and all neuromuscular and neuroglandular junctions in the parasympathetic division. So that's that slow division of the afferent nervous system. So everywhere, they're literally everywhere. And the idea here is that the action potential reaches the axon terminal. Calcium ions enter the axon terminal. That's a key part that I missed during my test taking. And you can see that here, figure 1216, slide 2. That's what kind of begins this neurotransmitter release. And acetylcholine breaks out of the vesicles via exocytosis. As it reaches the postsynaptic membrane, it perpetuates the action potential, and then the excess acetylcholine is removed from the synaptic cleft by, I think it's acetylcholinesterase, is going to clean up. So a lot of it, sometimes it's not all used. I think only like 50% of the acetylcholine is actually used, and everything else is recycled. Yeah, only half of the acetylcholine release reaches the post-terminal. All right. So that's pretty, that's pretty much it. We have these chemically gated cation channels here, and then the sodium can continue to propagate, and we can go to the next neuron if needed. Some of the more neurotransmitters that we have, we can classify them as excitatory or inhibitory, and that's going to depend on the receptor more than the neurotransmitter itself. Certain neurotransmitters can either be excitatory or inhibitory, so keep that in mind, all right? Acetylcholine, even though we just looked at it in an excitatory fashion, can also be an inhibitor, and it actually does that in the heart. That's because of the specific receptors there. We also have these biogenic amines. First one here is norepinephrine, which is a hormone that we know is secreted in the adrenal glands. It can act as a pressor because it's given during cardiac catheterizations under the name Levophed. It's also known as noradrenaline, and its synapses are known as adrenergic. And you would think adrenergic is adrenaline, which would be epinephrine, but it's not the case. So adrenergic, you have to remember, is norepinephrine, all right? That you are going to hear a lot in pharmacology. Catapres clonidine is a centrally acting adrenergic agent. So that's a good thing. You're going to pick up these as you go through pharmacology. Norepinephrine is typically excitatory, all right? Dopamine, and that's why it's a pressor. That makes sense. If it's excitatory, if it has kind of a go versus a slow effect, it's going to, I believe, beat the heart faster. Or technically, I think it's actually a vasopressor. So it's going to constrict your blood vessels and bump your blood pressure up, which definitely sounds excitatory to me. All right, so dopamine is up next. We've heard a lot of this one on social media, like those hits of dopamine, the old dopamine rush. It lives in the central nervous system in many regions of the brain. It can both excite and inhibit. And if the neurons that produce dopamine are damaged or destroyed, you can get Parkinson's. So dopamine is really important with your muscle tone. So if it's inhibited, you're going to lose kind of control over your muscles, hence Parkinson's and the tremors that can release. The interesting thing is that cocaine prevents the removal of dopamine from synapses. So it's kind of like opposite of Parkinson's. I would think you have like even more control over your muscles, which is kind of interesting. So it kind of gives off that high. So just remember, dopamine is the one that's involved with Parkinson's and cocaine. All right. Then we have Low Tide at Serotonin Bay, one of my favorite Wonder Years songs, Low Tide. This one is housed in the CNS as well. And this one is what we hear a lot with SSRIs. I'll see if I can name a couple from memory for you. So inadequate low tide production can cause chronic depression, okay? So enter the SSRI, which is the selective serotonin reuptake inhibitor. So this is going to disallow the reuptake of serotonin in these synapses here and allow more of it to be used. All right, so it's going to recycle less and use more of this serotonin because, again, low equals chronic depression. So we want more of this. So examples of SSRIs would be sertraline, which is Zoloft, escitalopram, which is Lexapro, fluoxetine, which is Prozac. We also have citalopram, which is Celexa. And then we have paroxetine, which is Paxil. See, I messed up. Cymbalta is that's an SNRI. Just confirm that. Yeah, good. Caught myself there. Okay. Celexa, make sure that one is. Yep. Paxil. Yes. Okay. So those are all SSRIs. Cool. All right. We also have some amino acids that can act as neurotransmitters. So we have GABA, which is gamma aminobutyric acid. Generally, it has an inhibitory effect. 20% of the synapses in the brain release GABA. Another pharmacology thing here, you have drugs like gabapentin or pregabalin lyrica, which are technically classified as anticonvulsants. And that is the same GABA that's involved there. Okay, so an inhibitory effect, GABA, that kind of makes sense to stop seizures. We also have neuropeptides. This is a really interesting one here. They're not technically neurotransmitters. That's why they're called neuropeptides, but they can affect like the release rates of, I believe, neurotransmitters. These are kind of like modulators. Oh, there we go. These are called neuromodulators. They're typically small peptide chains. An example of a key neuromodulator is called are opioids. Okay. There are three classes here: enkephalins, endorphins, and dynorphins. The textbook here really doesn't go into any detail. It just says the primary functions of opioids are probably to relieve pain, which is interesting. It means we're still researching that. They inhibit the release of something called substance P, which is involved with, it's a first response to extreme stimuli like pain or trauma. So morphine is a narcotic analgesic. It's the 140th most prescribed medication in the US. Opium or poppy tears is a dried latex, which is an emulsion from the seed capsules of the opium poppy. So before this opium poppy is fully bloomed, there's a little bulb, and you actually scrape the side of it, and this like glue comes out. And that's morphine. That's crazy to me. All right, it's called Papaver somniferum. Around 12% of opium is made of morphine. The poppy is grown for three purposes. This is totally not in the book, by the way. This is just something I thought was interesting. You can produce poppy seeds, like on your bagels, produce opium for the use by the pharmaceutical industry. I mean, what a difference in uses for the same thing. It's crazy. Or to produce other alkaloids like thebaine, which is paramorphine, and this oripivine, which are turned into drugs like hydrocodone, which you can hydrocodone combined with acetaminophen gives you Vicodin, and oxycodone, which is OxyContin. Poppy-based drugs are morphine and codeine, which you can combine with acetaminophen to create Cosette. Most of this comes from Tasmania, Australia. Who would know? So there you go. So they don't really go into a lot of detail. You definitely hear about releasing endorphins a lot. Definitely more to be learned from that section there. Okay. We also have dissolved gases like nitric oxide and carbon monoxide. Not a lot of detail in here in the book either. But if CO enters the bloodstream, if we breathe it in, like if you're trapped in the garage and your car is on, it binds with hemoglobin and causes carbon monoxide poisoning. Okay. Some neurotransmitters and neuromodulators have direct effects like acetylcholine, glutamate, and aspartate. Others use second messengers like G protein coupled receptors. And the last section here is individual neurons process information by integrating excitatory and inhibitory stimuli. So we have something called temporal summation, which is like filling up a bucket with 2 cups, but one at a time. And then spatial summation, where we fill it up with a bunch of cups all at once. That's really, just remember that. GABA can be used in an example of something called presynaptic inhibition. So this opens up voltage-gated calcium channels, which can reduce the amount of neurotransmitter release. That's pretty interesting. Oh, it inhibits the opening. Okay, that makes sense because calcium kind of starts the whole thing. So that's it. That's chapter 12, kind of in a nutshell. Hopefully that will help me commit a lot of this to permanent memory. I can now go back and listen to this when needed. hopefully somebody out there who's studying A&P chapter 12, hopefully some of this was helpful. But there you go. I will see you in the next one.